So that means we gotta storm to the end. So we only have what? 2 lectures this week and 3 lectures next week, right? Then we're ready to do it. So it's gonna come quick. Then you guys are free for what? 1 week? That's it? Right back in. Okay, so last time we were talking about a starting point to determine if evolution was occurring for any particular traits, either morphologically, physiologically, or behaviorally. So I gave you the Hardy-Weinberg principle, which you should be expecting to see again in one shape or another computationally on the examination. So make sure you do those computation steps, make sure you can wind your way through it. The model itself mathematically is not exactly the most exciting thing, because if you do everything right and that trait's not changing, what does it result in time and time again? The same thing. Same allelic frequencies, the same phenotypic frequencies over and over and over again, okay? The more interesting thing is what? If it does change. And so if it does change, that tells you that there's evolution occurring to that particular trait. So the model has 5 basic premises or assumptions to it, right? Number 1, what does it assume about the mutation of the gene? Is it occur? No mutation. What about the population size? It's going to be really big. Now what about that population in regard to immigration and immigration? There is none. It's isolated. What about the gene survivability or reproductive characteristics? It's basically neutral. And then finally, mating is random. Those are the 5 things that you would look at to basically decide what is driving the evolution of a particular trait if those phenotypic, if those allelic frequencies changed in this model. All right? To summarize it, remember there are 3 basic processes that typically drive populations away from equilibrium for any given trait. What are the 3 things? Natural selection, gene flow, which is the movement of alleles in and out of populations, and genetic drift, which is the random fluctuation of alleles due to sheer chance events. Okay? All right. We began talking about the sources of variation. Let's run through the basic premise again. Variation acted upon by what's the microevolutionary process? What is it? Variation acted upon by what? 5 steps. What are they? Variation acted upon by natural selection leads to genetic divergence supported by reproductive isolation, which can lead to speciation. All right? That's the microevolutionary process. So we started taking a look at the sources of variation. What's the ultimate source of variation? Mutation. Why? Because it creates brand new alleles. That's exactly it. What's the secondary source? Recombination. Does that mean it's not important? No, but it's just doing what? It's just reshuffling the deck of alleles that are already there. But it doesn't mean it's not important, because the amount of variation driven off that is astounding. All right? So mutations give you something unexpected behaviorally, morphologically, or physiologically. And so big mutations that are talking about deletions of material or duplications of material or inversions or translocations, what are those referred to as? Those are chromosomal mutations, and they're large things for animals. Are they good? They almost always result in what? Death. Plants have a better chance of actually handling that without killing the organism itself. The much more common mutation, however, deals with an addition, subtraction, substitution of a nucleotide base. What would that be referred to as? That's a point mutation. They're quite common. They can be what? In terms of adaptive, maladaptive, or neutral, depending on where they fall. Lots of times they're completely neutral, primarily because they fall where? In non-coding regions of DNA. They can be troublesome sometimes if they fall at the start or the end of something that's initiating an amino sequence. Right? That can be problematic. All right, so they can be silent. They have no effect. They can reduce the efficiency. What would that be referred to as? A missense. Very good. Or they can create a non-functional protein. What's that? Nonsense. Very good. All right, if a mutation gives you an advantage in survival and reproduction, it's considered to be what? Positive, adaptive, yes. If it decreases it, maladaptive, and no effect, neutral, very good. All right. Are mutations rare events? They are. However, even though they're rare events, they're still the stuff of evolution. Why? They're rare events. How can they basically give us all this variation? Because of evolutionary timescales. We're talking 3.8 billion years of evolution, and we also know that each gene is eventually going to do what? it's going to mutate sooner or later, and there's tons of individuals typically within any given species. So the number of individuals, knowing that there are mutations, and the immense amount of time for that to occur, allows all this stuff to build up over time, all right? It's the raw material for evolutionary change. We talked a little bit about recombination. If I was to say that this was sexual reproduction between two genetically similar individuals, what would that be referred to as? Inbreeding. Is that a good idea? That's a bad idea. What does it increase? It increases the genetic load, which is the tendency to put those recessive traits together and express negative maladaptive traits. Also, what does it decrease? The amount of variation. Why is that a problem?
Can't deal with environmental change.
Because you're not as well adept at dealing with environmental change from a population perspective. Remember that the variation is an arsenal against environmental change. The smaller it is, the less able we are to cope with that. That means if you got a disease and everybody's genetically similar, what can happen? Everybody dies, you go extinct. If there's variation in that population in terms of resistance, certain members of that population are going to survive. You reach a thermal maximum, everybody will die. But if there's variation, what's going to happen? Somebody's going to exist and be able to repopulate that population by passing on those traits with a higher frequency. So that's the advantage of carrying all that variation. Crossbreeding then is exactly the opposite. Sexual reproduction between two genetically what? Different individuals of the same species, of course. And that's going to do it to the genetic load, decrease it. That means less harmful things are expressed. And what's it going to do to the variation? It's going to increase the variation, which means you have a better chance to withstand any kind of environmental change from a population perspective. We talked a little bit about sexual selection. Traits are selected simply because in sexual selection, why are traits selected? In what regard? It's just because one or the other prefers them. That's all. So remember, it's always a balancing act. So I can decrease my survivability as long as I do what? Increase my reproductive output. And so often what you see with sexually selected traits is often a decrease in your survivability, but an increase in your reproductive success, and that can basically balance out. And this can occur both intra and interspecifically. It often results in what characteristics among the difference between a male and a female? Dimorphism in either behaviors, morphology, and physiology in some cases, right? Very common in mammals and birds. Individuals look different. We talk about hybridization? Yeah. So hybridization is a form of what kind of breeding? It is crossbreeding, but it's not within a species, it's what? Between two different species. That hybrid often has problems, though. In the case of the mule I gave you, what was the problem? The male was what? And the male was sterile and the female was fertile. That's often a problem with hybrids. We're going to talk about that momentarily. Why hybrids, there is a problem when you cross species like that. It often doesn't work. It doesn't end up with viable offspring. All right, so sexual reproduction between two different species. And I think we started, did I finish talking about the balanced polymorphism? Sickle cell anemia? Did I? I thought that's where I stopped. Let's start there. We'll review. It's been a little while since the holiday. All right. So remember, balanced polymorphism simply means you're maintaining two or more alleles. And in that combination, they persist in the trait. The trait persists in the population over time. That frequency is greater than 1%. You can shift around, but you constantly return to that initial value. So I gave you the example of sickle cell anemia. And so in this particular case, there's a mutant allele. It's HBS, and it gives you this sickle cell shape. It's problematic because what's the problem? What happens when it's moving through your circulatory system? It gets stuck. That's exactly it. And it causes stroke, seizure, cardiac arrest, all kinds of problems, right? And so you get stuck in the small vessels all around your body, causing a whole series of nightmarish problems, and actually killing you eventually. So HBA is the normal red blood cell shape, which is biconcave in nature. What was the shape of the cell again? What's the function of that biconcave? It doesn't have a nucleus? It increases the surface area for the transport of gases more efficiently, right? Again, if I have sickle cell, can I carry the gases efficiently? So it's clogging up and it can't carry the gases effectively. So that means if I was to run and I have normal blood cells and I'm in a race against someone with sickle cell, who's going to win that race? Probably me, because I'm carrying much more gas and a much more efficient man, all right? So it's problematic. Now, from our perspective, we would think that this trait is what? Adaptive, maladaptive, or neutral? From our perspective here in America, it's quite maladaptive because it's causing all this negative consequence for you, right? It's going to shorten your lifespan. It's tough to kind of get around without sucking wind all the time. It's not the easiest thing to live with, and your lifespan is going to be shortened. If your lifespan's shortened and you're cut off before you can reproduce, then you're not going to be able to pass anything on. Your fitness is decreased to zero. So it's a problem. However, when we look at places around the world, we see that it's found at frequencies much higher than we would expect for a maladaptive trait. And it's really quite unusual. And so when you take a look at the tropical band, the first thing you see is this big swath of individuals who bear this in the heterozygote form, which is basically HBA, HBS. In this particular case, you have both types of blood cells that are being expressed. You have the sickle cell and you have the normal cell. So they're both in your system. It's definitely not as good as having just regular blood cells, right, in terms of what we perceive as negative consequences. But they're living, and they're in high density in relationship to what they should be if it's a maladaptive trait to carry that particular allele. So what's your basic question? Yeah, why? Why would you have it? All right? So, I'm sure there's a way to get the screen low. Oh, there we go. I wished, and I made it happen. There you go. Okay, so again, it's much higher than you would expect. Up to 1/3 of the population is heterozygote in this particular condition. Well, the first thing we see is, well, why? And that's our basic question. So when I investigate this particular question, the first thing I see is that it falls right on a band of the globe where malaria is very, very high. Okay? So I see a propensity to have this particular allele where I find malaria. When I take a look at the consequences of disease, HBS individuals often die with the sickle cell itself, all right? Something about the condition is going to kill them sooner or later. HBA are perfectly normal. However, they die at a much higher rate wherever I find malaria. And then when I take a look at the HBS, those heterozygotes, they make about 1/3 of the human population in those areas and die at a less rate than people who do not have the allele. Well, that's pretty curious, right? Because we consider the HBS allele to be maladaptive and negative, shortening your life. However, when you have that particular allele as a heterozygote in malaria-rich conditions and areas, you survive at a higher rate than people who don't have it at all. Why? Any idea? Anybody ever had malaria? It is a crappy disease. I've seen it many times, and I've been infected with something similar, and it's a brutal disease. Crushing fevers, slam you up to the point where it feels like your blood is boiling, lose control of yourself, don't know who the hell you are, because your blood is so hot, body, misery, then you crash down, then you go up and down and up and down, and it's because of the nature of the parasite, because it ruptures your red blood cells. Pretty brutal business, right? Not exactly the nicest thing to go through. This is actually urine from a patient. You can't see it because the lights are pretty crappy, but one of the symptoms is because of the rupturing of those red blood cells, you often see blood in the urine. So it can get pretty nasty. This is a victim over here. There's the plasmodium, which is the disease itself. And here's the vector. You can see how it's in the red blood cells there. That's why it's so tough to get at. Because when it's in you, right, in your cells, that's a really tough parasite to get rid of. When it's between the cells, it's easier. But when it's right in them, it's tough. And so this is the Anopheles mosquito, the female. She's the carrier of this thing. And so, the way it basically works is she's going to bite and spread this disease around. And then you're going to go through the symptomology of the disease and go through the life cycle and have all that unpleasantry. There's a series of medications that can prevent it. They're expensive, though. If you travel to the tropics, you're often given them. So I used to be a tropical ecologist, and I had to take all that stuff, and I hated them all. They're miserable. They do all kinds of weird things to you. The anti-malarial medication that I have used to screw with me. It used to give me terrible nightmares. And then the worst part about it was I remember there's somebody telling me, yeah, you gotta be careful. You'll feel like you don't have an arm or a leg or something like that. What the hell are you talking about? And I remember I was driving and I lost, my whole arm just disappeared. I couldn't feel anything. And I was just like, whoa. I was like, whoa, what the hell? I was having a heart attack or something. It was just the medication. It did weird things to my nervous system, right? But I didn't get malaria, and malaria was scary. So I guess it was a trick. And they made you drink this terrible water like quinine. Have you ever had quinine water? Oh God, it's miserable. It's like this really bitter kind of tonic water. But they're malaria. If you go about with malaria, I'll take anything over malaria. I'll have missing limb, whatever it takes to not get it because it's such a nasty disease. So in this particular case, this combination of alleles is sustained at much higher densities than you would expect by sheer chance alone. So over 300 million people are a year infected with malaria. Malaria has actually influenced the evolution of the human species. Okay? It's a powerful disease. So the frequency of the heterozygotes are higher, the malaria incidence is highest, and HbA, HbS heterozygotes are more likely to survive. So why do they survive? If I'm perfectly normal for the allele type and I'm homozygous and I'm HbA, HbA, and I get malaria without treatment, what will happen to me? I will die. Okay? So without modern medicine, I will most likely die. However, if I'm HbA, HbS, I most likely will survive. It will be unpleasant, but I most likely will survive. Now the question is why? That's a great idea. That's not quite it, though. There may be some of that. That's actually a great piece of research to look at. But that's not the mechanism that we have today. So what do you think it is? Well, the way that this thing works, remember, is it's going through these cycles of boom and bust. Whenever it busts, it moves around. How's it moving? It's moving how? With your blood. Your blood moves quick. And everybody had a CT scan or something like that, and they gave you the injection, and you can feel it move around? You ever felt that thing? That's a strange experience, right? They'll inject that dye into you. You can literally feel the dye go right from the injection point through your heart and down through your body. It works its way around. It's kind of an unpleasant, weird experience, but it's fast. And so your blood is moving very quickly. So in a normal person, your blood's moving really fast because the blood cells are what? They're normal and they're round and they're flowing through the pipes very cleanly. However, if I have sickle cells, what's the rate of motion? It slows down. Well, this thing is developed and evolved for dealing with human beings who are moving blood at a high rate. That's how it moves around and completes its life cycle. So if you slow that down, what does it do to its life cycle? It slows it down too. And if you slow down its life cycle, what can your immune system do? It has the time to fight it. That's exactly right. And so what you see in this particular case is having this allele gives you a higher survivability in regards to being homozygous dominant for the normal red blood cell shape, which is quite interesting. So here's the situation. We consider this to be a maladaptive trait. However, if I live in sub-Saharan Africa and I have this, is it maladaptive anymore? It is not. It is what? It is an adaptive trait to be heterozygous. It's not adaptive to be HbS, HbS. Why? Because I'm going to die from the sickle cell, right? But if I'm HbA, HbS, that's going to give me survivability against malaria, which is a constant threat in my everyday life. It's a driver, an evolutionary driver. So therefore, I see a balanced polymorphism between these alleles, which I would not expect by sheer chance events. All right. So again, the non-mutated cells maintain the body's function. This is in a heterozygote, right? We have non-mutated cells maintaining the body's function, and the mutated cells interfere with the circulation, which interferes with the life cycle of the parasite. In the tropical regions, selective forces have been in operation for over 200 plus years. Okay? So what we're seeing is a complete change in human evolution based on a parasite. You'd be amazed at what diseases can do in terms of the influence on history. So what was the biggest flu to ever hit? That decimated the most human beings that we know of? Spanish flu was one of the biggest ones, right? The Spanish flu directly had an influence on the outcome of the war. Without it, we're not sure that the Allies might have won that war. Isn't that interesting? So that disease was actually spread to German troops who were winning the war at that time. And it was tracked down to tunnels in France, underground tunnels, where French soldiers who were sick with the flu transferred it to German soldiers who then brought it back to their ranks and it decimated their ranks, weakening them up sufficiently enough to end up with victory from the Allied forces. Isn't that crazy? So again, flu influencing the history of human beings. Pretty interesting when you think about it. There's lots of examples of that around the world. You can trace back time and time again to see those occurrences. Microbial world influences our basic evolution all the time. All right. Let's talk about the types of variation. So I've spelled out the sources of variation. What are the sources of variation again? Primary is mutation. Secondary is recombination. Now we're going to talk about the types that you find. And so the primary type, the most common type, is normal variation or continuous variation. It is quantitative by nature. It is things like height and weight, any basic trait that I can pull out. So have we ever done this before where I've asked you guys to raise your hands for height? Did I do that? So when I did that, what was the low end? I can't remember, like 4'10" or something? And the high end was what, 6'5" or something like that? I can't remember. Then there was a bunch of people in the middle. Did I do that in here? Yeah. Okay. And so we had people on one extreme on the other, and then a bunch of people in the middle. And if we plotted it out, what would it look like? It was a nice hump-shaped relationship, a unimodal curve, okay? So anything along a continuum like that has a tendency to plot out as this very nice hump-shaped relationship. So that's the first type of variation we see. I can do that for anything. Space between your eyes, length of your pinky finger, length of your femur bone, your humerus, whatever it might be, they're going to plot out like this. However, there are some traits that don't fit along the continuum, and it's either this or that. Remember, they're discrete traits. And so things like eye color, a nice example, they're blue, brown, black, because of the way the lights switch on. There are some blending, but it's very distinct. Blood groups is a great example. So blood group, if you're either A, you're B, you're what? You're AB or you're O. That's it. There's no in between. Okay? So if we're A and I gave you B, is that good for you? No, if you're going to have a big immune response to that, it's not going to go well for you. Okay? So they're found in discrete groups. It's this or it's that. And then the last one is ecogeographic variation. So what does that look like when it's plotted? It comes out as distinct humps, right? Not this nice clean one, but it's like up, down, up, down, up, down, up, down. Then we have ecogeographic variation, and that's variation along some kind of climb, ecologically or geographically. So it would be like, how does a trait vary with water from low to high? How does a trait vary with temperature from high to low? How does a trait vary from north to south or east to west? And so typically when we plot that, they have a tendency to either go up or down in terms of the measured trait and the number of individuals. They don't have to be linear. They can be curvilinear, but they have a tendency to trend either up along the gradient or down along the gradient. Okay? So plant morphology would be a nice example. This is what this little diagram here is showing. Basically, it's saying that when plants have a lot of water, they're real tall, and as the water supply gets smaller and smaller, the plants get smaller and tinier and tinier. Yes? Eye color is kind of considered to be a polymorphic trait, but the more we learn about it, it's more continuous. The problem is you're kind of considered to be brown or blue. It's rarely you say I kind of have brownish blue eyes. Skin color is much more along the continuum. It really is. That one's kind of tricky, and I'm not 100% how to place it, but I would say it's more continuous than anything. I don't think that's been found as a discrete unit at all, because I mean everybody has trends, even within distinct, like if you have white skin, it's olive skins, there's all kinds of different coloration patterns of that. I just don't see it so much. All right, so those it. Normal variation, polymorphic variation, ecogeographic variation. Again, normal, along a continuum. Polymorphic, distinct, discrete units, and then ecographic climbs up, climbs down, geographic or ecological gradient.